Brazil, late 2014. Doctors noticed a disturbing trend. An increasing number of babies were born with abnormally small heads, a condition known as microcephaly. The spike in microcephaly was linked to a virus called Zika that is spread by mosquitoes.
I just never dreamt, as an epidemiologist of infectious disease, that I would see a congenital transmission by a virus that would do such a great damage and would be transmitted by mosquitoes.
Throughout the Americas, mosquito bites are tied to many viral outbreaks. In the months before Zika spread in Brazil, chikungunya, a tropical virus that causes terrible joint pain, invaded the Western Hemisphere, infecting almost 1 million people. And West Nile virus sickened more than 2,000 victims in the US, with symptoms ranging from fever to paralysis. To stop these outbreaks, scientists have turned their attention to fighting the mosquitoes that carry these diseases. And one of the weapons they are using is genetic engineering. In South America, the Zika virus is spread by a mosquito called Aedes aegypti. Other diseases, including dengue, chikungunya, and yellow fever, are also spread by this persistent insect.
Aedes aegypti is the cockroach of mosquitoes. It lives indoors and outdoors. It bites daytime and nighttime. It develops resistance to insecticides easily. The eggs can overwinter for a year. To control it, we have to kill the adult mosquitoes, the larval mosquitoes, both inside and outside.
Health officials have used sprays and screens to protect people from diseases spread by mosquitoes. But since 2002, scientists at a biotech company called Oxitec have been investigating a more direct approach. A line of genetically modified Aedes aegypti mosquitoes called OX513A.
The goal of this procedure here at Oxitec is to use our mosquito, OX513A, to reduce the population numbers of the Aedes aegypti mosquito in the wild.
When released, these mosquitoes reproduce with wild mosquitoes and cause their offspring to die. Although there are concerns about this technology, authorities have allowed limited releases to study its efficacy and safety. So how were these genetically modified mosquitoes produced? The process involved engineering mosquitoes with 2 new genes.
What you do first is create or synthesize the DNA you want to insert. So in our case, we use the 2 genes that we inserted, the lethality gene and the fluorescent marker.
The genes were inserted in the genomes of mosquito eggs. The lethality gene makes the developing mosquitoes dependent on the antibiotic tetracycline, which is fed to them in the lab but is not available in the wild. The fluorescent marker gene produces a protein that glows red when exposed to light of a certain wavelength. It tells scientists which mosquitoes have the lethality gene. After the first genetically modified mosquitoes grew into adults, scientists bred them in the lab. These mosquitoes passed on the genetic modification to each new generation, giving rise to a colony of genetically modified mosquitoes, all dependent on tetracycline.
So this is our female room. You can walk in. In these big cages, what we have is our egg-producing colony. In total, about 12,000 females per cage. And they are here to produce the egg. So our genetic modification has been done in 2002. A stable inserted in the genome of every single of these mosquitoes, which are here in this room. They lay their eggs in these pieces of paper, all the black little dots our eggs that have been laid.
When scientists plan to release genetically modified mosquitoes into the wild, they collect eggs from this colony. They place the eggs in water, where they hatch into larvae, and then grow into pupae. At this stage, technicians separate the males from the females by size.
These are a female, and that's a male pod.
The male pupae are significantly smaller than the females. Feeding them tetracycline, the antidote to the lethality gene, keeps the mosquitoes alive as they grow into adults.
So we give an antidote in the lab to the mosquito so that they don't die. And then we go release our male into the field.
Male mosquitoes don't bite people, so they will not spread disease, but they live long enough to mate with female mosquitoes.
And then life becomes exciting for them. They get to do what they're best at doing, which is searching females, copulating with those females. And our males will deliver the lethality gene to their children. And in the wild, the babies do not come in contact with the antidote. And there they die.
Well, here in this van, we can fit almost 800,000 of male mosquitoes. As we go driving through the area, every time the app beeps, we just open one of the spots in here and the mosquitoes will fly around and do their job.
Once released, the genetically modified male mosquitoes mate with wild females. The male carries 2 copies of the genetic modification, so all the offspring inherit a copy of the lethality gene. The wild offspring will not feed on tetracycline, the antidote to the gene, and they will die before reaching maturity. Scientists monitor how the technique is working by capturing larvae and determining what percentage have the telltale red fluorescence. Studies have shown that with repeated distribution, the wild mosquito population of a village or neighborhood can be cut by 95%. If the technique continues to prove effective and safe to humans and wildlife, it could be applied to other species of mosquitoes and help stem the spread of many dangerous diseases.